let's begin with some simple biology. Think of your body as a protein-producing machine. Muscle cells make muscle proteins called actin and myosin. Skin cells make skin proteins called collagen and elastin. Stomach cells make stomach proteins called enzymes. Your immune system makes immune proteins called immunoglobulins or antibodies. Your eye cells make eye proteins called keratin. And every single cell in your body, except red blood cells, make proteins. And the expression of proteins is the expression of life, because proteins are responsible for the structure and the function of your body. In order for a cell to make proteins, a gene has to be stimulated or regulated. So what are genes then? Think about genes as a library of potentials. And so there was a myth after the human genome was discovered that genes create disease. But if you study the research, less than 1% of people on the planet are born with genetic conditions, early onset genetic conditions like Tay-Sachs disease, sickle cell anemia, type 1 diabetes. The other 99% is created from lifestyle, behavior, and choices. So in order for a cell to make a protein, a gene has to be regulated. So then, if you understand then that genes don't create disease and the expression of proteins comes from genes being regulated, then what is causing genes to become regulated or stimulated? Well, the latest research in the field called epigenetics says that genes don't create disease. It's the environment that signals the gene that begins to create disease. But it begs the question, how can two factory workers working side by side, both exposed to the same carcinogenic chemical, one gets cancer and the other doesn't? Surely there must be some internal order in that person's body that's greater than the conditions in their environment. And in a sense then, the inner environment of the body is still the outer environment of the cell. So then, it's not genes that create disease, it's our reaction to the environment, or better yet, how we begin to look at the environment that influences our emotional state. And it's our emotions then that have a key factor in sending signals to cause genes to upregulate or what we call downregulate. Now, genes are like Christmas tree lights. They're turning on and off all the time. But in a sense, they're not turning on and off. They're getting regulated, or they're getting instructions from the environment outside of the cell. And when they get the proper instructions, when the gene gets upregulated, it makes a healthy protein. When the cell is getting information to cause it to contract or limit its metabolic productions, it begins to downregulate, and it starts making a cheaper protein. So then, if the environment signals the gene, and the end product of an experience in your environment is called an emotion, is it possible then that you can signal the gene ahead of the environment? We learned in previous episodes that the stronger the emotion that you feel from some experience in your life, the more altered you feel inside of you, the more you narrow your focus on the cause, and the brain freezes the image, and that's called the memory. And we said that people think neurologically within the circuits of those past experiences, and they think chemically within the boundaries of those emotions. And how you think and how you feel creates your state of being. So in a sense, highly charged emotional events can possibly signal genes that begin to cause the body to change. So then, if a person is waking up every morning and searching for the familiar feeling that they call themselves, the moment they get in touch with that feeling that's familiar to them, and those feelings are influencing their thoughts, and those thoughts are creating more of the same chemicals that create the same feelings, then if they're living by the same emotion, it makes sense they're stimulating the same genes in the same way every single day, and now they're headed for a genetic destiny. Because there's no new information coming from the environment that could possibly begin to select or instruct new genes. And in fact, if you look at two identical twins, both sharing the same genome, why is it that one dies at 54 and the other dies at 86? Obviously, it's their interaction in the environment, the choices that they make, the behaviors that they demonstrate, how they respond to stress, how resilient they are to return back to homeostasis. And in fact, it's their interaction in the environment that's selecting and instructing gene expression. Now think about this. When they began to map the human genome, they said, okay, the body makes about 100,000 proteins, which means there should be 100,000 genes for each one of those proteins. We have 40 genes that are called regulatory proteins or regulatory genes that assist in making those proteins. So now 100,000 plus 40,000 makes 140,000 proteins, and there should be 140,000 genes. But when they mapped the human genome, they found out that we only express, as human beings, 23,688 genes. So then how is it possible then that we have more proteins that make up our body than we have genes to make them? Because on one single gene, there can be thousands of different variations that begin to cause the cell to begin to make different types of proteins. So then the expression of proteins is the expression of life. And so if people are living in the routine of the predictable future, or they're memorized to emotions of their past and they can't think or act greater than how they feel, then those series of events in their lives has began to influence their gene expression. Then once the gene has been signaled and the person is constantly living out of homeostasis or balance, that imbalance becomes their new balance, and now the internal environment of their body is sending specific signals to the cells, and the cells are getting the same instructions, and the same instructions produce the same proteins, and now the person begins to experience ill health or disease.